which was very impressive. So welcome to this perioperative allergy session. Are you up for a challenge? My name is Alison Brereton. I'm an anaesthetist and anaesthetic allergy specialist from Adelaide. And today I'll be chairing this session, which starts with a presentation by Paul Sadlier, followed by a panel discussion which will be stimulated by your questions. After our presentation, we'll invite you up to ask questions by the microphones on the floor. And we would love to know your name and where you are from before you pose your question. You can also send in questions via the event app, and we will get to as many of those as we can in the time available. I hope you're all experts in using the app to put your questions in by now, because you've all had 3 days practice. So let's get underway. I'm privileged to introduce our first speaker today. Dr. Paul Sadlier is a cardiac anaesthetist who has been investigating perioperative anaphylaxis at the Western Australia Anaesthetic Drug Reaction Clinic for over 10 years. He is active in allergy research and education, and most recently has focused on the prevention of life-threatening perioperative reactions. He is someone who always has another perspective. Today he's going to talk to us about a cure for severe intravenous anaphylaxis. Thank you, Paul.
Thanks, Alison. Thanks, everyone, for coming. So, perioperative allergy is sort of an intersection between two specialties, between immunology and anaesthesia. And I think the different specialties bring different things. In immunology, they're primarily focused on investigation and diagnosis and the secondary prevention of further reactions in patients. Whereas in anaesthesia, we're focused on the treatment of anaphylaxis, of perioperative anaphylaxis, and also the primary prevention of anaphylaxis in our patients. And so primary prevention, what does that mean? For us, it's things like trying to get Fulcadine banned to reduce the risk of neuromuscular blocking agent anaphylaxis, which we've done. It means things like taking a drug history and having that part of your team timeout, even though we know that the drug history is often inaccurate or doesn't really reflect the biological truth. And we choose low-risk drugs or low-risk allergens. So we might try and choose an alternative to suxamethonium that has a lower risk of anaphylaxis for most of our patients, unless it's indicated. There used to be a fourth way we could prevent anaphylaxis, and that was called the test dose. But then NAP6 came along about 5 years ago and really ended that. So, whoops, different button. So what did NAP6 find? So NAP6 was the year-long survey of the UK of cases of perioperative anaphylaxis. And there was a few things which I found remarkable about what they found. And the first one was that they reported that a third of anaesthetists in the UK give a test dose. And this is much different to my experience in Australia. Not nearly that many people give a test dose in Australia, or so I thought, although I often do give a test dose. And I know the logic is against it. So also with NAP6, the majority of cases of anaphylaxis in the UK were caused by antibiotics, and they noted that these are the ideal agents for giving a test dose. The second most common cause was neuromuscular blocking agents. So in NAP6, in their report, they give the rationale for giving a test dose. Now, the rationale is that anaphylaxis isn't an all or nothing response. It's a dose-related phenomenon. And the intention is, in giving a test dose, that you're giving a small dose of the drug that will, in a patient that's hypersensitive to it, elicit some mast cell degranulation that you can recognise before you've given enough of a dose that they have full-blown anaphylaxis. So if you do have a small amount of mast cell degranulation to your test dose, you would treat it and withhold further allergen. You've prevented harm to the patient, and this is a success story. However, they found that the doses that they observed being given were very large, and also that there was insufficient time between the test dose and the rest of the dose for a reaction to be recognised and avoided. And their one recommendation was that you should give antibiotics 5 to 10 minutes before induction. mainly so that it's easy to recognise what the trigger was that caused the anaphylaxis. But I think also if you give an allergen in an awake patient, the physiology is much more resistant to, say, cardiovascular collapse, as opposed to if you've got someone that's induced under anaesthesia with positive pressure ventilation. There are lots of reasons why the severity of anaphylaxis would be a lot worse. So what did they find? They found they had 266 patients that were referred over that year that had anaphylaxis, which I might add is a very low rate compared to Australia. Of those 266, 235 got an antibiotic, and of those 235 that got an antibiotic, 92 of those subsequently were diagnosed at the clinics as being due to antibiotic anaphylaxis. The other ones were muscle relaxants or gelifusing or something like that. They did present some of the cases. So let's look at the cases, which is on the right of screen. So first one's an example of a patient getting 120 milligrams of coamoxiclav IV, and then 10 minutes later getting the full dose for which they had anaphylaxis to. The next patient had 300 milligrams and got the full dose 1 minute later, and then they developed a PEA arrest. The third case, they were given 180 milligrams of coamoxiclav as a test dose, and they had anaphylaxis to that test dose. So these are examples of these are the three examples they've shown of people using test doses inappropriately, or perhaps why test doses don't work. So of the 18 people that got a test dose that ended up having antibiotic anaphylaxis, 10 of them had a reaction to the test dose itself, and 8 of them had a reaction to the rest of the dose, the full dose. And they noted that the test dose was between 5 and 30% of the full dose. Now, this is a little bit curious because if you look at any allergen, and the best examples of allergens that have been investigated for the provocation threshold are food allergens, because it's a massive industry. You know, whenever you pick up some biscuits from the supermarket, they'll have in them whether or not this product, they'll state things like this product may contain traces of nuts. And what they define as traces has been determined by by sampling of population thresholds for those particular allergens. So they will define a particular threshold, say 0.01 milligrams of peanut protein, and they will decide that maybe 5% of the population will react to that threshold, and so that's what they'll pick as the threshold for which you have to label your product if it's more than that amount of protein in that. So a lot of money has been spent on creating those relationships between number of allergic people in the population reacting to a particular dose. What we know from those is that they're distributed over many orders of magnitude, and I'll show you some graphs later. But what I can tell you now is that we would expect far more patients to have reacted to the test dose than what NAP6 observed. We would have expected about 70 or 80% of the patients being given between 5 and 30% of the full dose to have reacted to those test doses. So the question is, where are those patients? And if you look at the numbers that they've reported, so NAP6 just reported absolutely test dose is a waste of time. But then if you look at the numbers, they had test doses given to 82 patients, and of patients that reacted to an antibiotic, only 18 patients received a test dose. So what that means is if you create a 2x2 table, you'll see that if you gave a test dose, the likelihood of the patient in the clinic testing positive to an antibiotic was only 18%. If you didn't give a test dose, the likelihood of a patient testing positive to an antibiotic was 55%. And this is a remarkable difference. And at the time you gave the test dose, you didn't know at the time you gave the test dose, you didn't know they were going to have anaphylaxis. At the time you referred the patient to the clinic and told the clinic whether or not you gave a test dose, you didn't know what the trigger was. So this is a remarkable difference. Now, there's a few explanations. One explanation could be people that are having anaphylaxis to your muscle relaxant, you might not yet have realised that anaphylaxis is occurring. But maybe you feel a little bit uncomfortable, and so when you then give your antibiotic, you do give it with a test dose. But I think the more likely explanation is actually that a lot of these test doses worked. The only patients that ended up in the clinic were the patients that got really large test doses and had anaphylaxis. The patients who got really appropriate, very small test doses probably just developed grade 1 anaphylaxis or a grade 1 hypersensitivity, which was not allowed to be referred to NAP6. So the anaesthetist probably realised that after they gave the test dose of the antibiotic, they got a few wheals and a rash. They didn't give any more. They treated it. They didn't refer it, told the patient they were allergic to the antibiotic, and harm was avoided. That's my hypothesis, and it's only a hypothesis. I might be wrong, but that seems like a reasonable explanation. And it's certainly the complete polar opposite conclusion to what NAP6 came to. So NAP6, what it does tell us, without a doubt, is that there is a demand for test doses, or there is a demand. Anaesthetists want to prevent anaphylaxis. And I might say, as an aside, if you have a patient with a beta-lactam allergy, which is almost 10% of your patients, or if you're in an ICU population, it's actually 20% of your patients report a penicillin allergy. If you don't give them a beta-lactam antibiotic, you will increase their anaphylaxis risk because the risk of those particular patients having allergic reactions to the alternatives like vancomycin, clindamycin is higher than if you were to give them a beta-lactam. And also you'll double their surgical site infection risk because you're giving a less effective antibiotic. So there's lots of reasons why we need to decrease the risk of suffering anaphylaxis. So the aim, if we were going to give a test dose, it absolutely, I'm the lazy anaesthetist there is, it absolutely has to be convenient and easy. I think we should be able to prevent 90% of cases of severe anaphylaxis. And then we'll all be able to say we're 100% sure that we've given the safest possible anaesthetic possible. Because, I mean, I've been working in a clinic for 10 years. I see people who come in with broken ribs. I don't see the people that die, but I see the people that come in with broken ribs with cancelled surgery. And they always say to me, is there anything that could have been done? And I always say, no, there's nothing they could have done. There was no way of knowing. But you all want to make it easier. So this is a teaser. This is my colleague Russell Clark, who also works in the allergy clinic, and this was last week. And this is just a trial of setting up this test dosing device. So what it is it's a syringe with 2 chambers in it. In one chamber is the drug, and in the other chamber is some saline. You put it in the syringe driver and you start the algorithm, and it will administer your antibiotic over half an hour. What it does is it dilutes the drug as it gives it. So initially you're giving an incredibly dilute concentration of drug. So at the moment you can see Russell's just putting the drug, which we've put some blue dye in so that you can see it. He's injecting the drug into the drug chamber. He's doing it very slowly because we're videoing it, and it's only the second time he's ever done it. So it's not too difficult to do. He's going to turn it upside down and just suck the bubbles out of it so that it's completely debubbled. He's going to put the cap on the back, which is the plunger cap. He's getting heckled by one of the nurses behind him at the moment. This would be ideal for all ward and IV infusions on the ward. Things like chemotherapy drugs, during the course of a platinum chemotherapy course over weeks, almost everyone develops anaphylaxis at some point, and so they often have to desensitise their patients. So this would be good. So he's injecting the saline into the dilution chamber, and you'll debubble that. And then that's finished. That's set up, ready to go. Chuck it into the syringe driver with some minimum volume extension tubing on it. And then you just select the algorithm, which is just in a drug library, like you would for any of the... So the program will run over half an hour, or this particular one. You can select how long you want it to run for. I think I'm going to... This part is just priming the minimum volume extension tubing. So as you prime it, you'll be mixing the drug. The initial drug that goes towards the end of the tubing will be very dilute, so it'll have a very light blue colour. And then closer to the syringe, it'll get darker and darker, and that's what I'm attempting to show you here. So as the infusion continues over the half an hour, it gets more and more concentrated, but also the rate of the infusion changes. So NAP6 gave recommendations that we should start all the antibiotics before we start anaesthesia. Now, why do we need to change? We don't like change. No one likes change. Anaphylaxis is the most common cause of death related to anaesthesia. When I tried to present to the Clinical Trials Network last year, someone disagreed with me, so I've looked up every review in the last decade and a half, and they all say that anaphylaxis is the most common cause of death related to anaesthesia. And if we could give our drugs in a safer way, we should, but I mean, that's a bit emotive. I shouldn't say this. I'm just going to give you some reasons why we should. Anaesthetists are extremely good at prevention. That's our sort of reason for being. We prevent crises rather than treat them. We pre-oxygenate people, even though the risk of a can't intubate, can't ventilate death is about 1 in 32,000, so the number needed to treat is far in excess of 32,000. We use BIS on patients for awareness. Number needed to treat for high-risk patients is just over 3,000. So for low-risk patients, it's well over 30,000 number needed to treat to prevent a case of awareness under anaesthesia. What is the burden? If we give an IV infusion or an IV injection of penicillin, 1 in 3,000 administrations suffer anaphylaxis. 1 in 10 in the general population report that they're allergic to beta-lactams. 1 in 20 ICU admissions. And so we'd estimate that just in the whole hospital population, there's about 70,000 inpatient IV reactions to antibiotics in Australia. And NAP6 found that the risk of anaphylaxis with teicoplanin, which I hope doesn't, become very common in Australia, although one of the hospitals in WA started using it, the risk is only 1 in 6,000 administrations for anaphylaxis. And then, as I mentioned, some of the platinum chemotherapy agents have a much higher risk. In NAP6, a third of patients experienced harm after anaphylaxis. And we know that from some research we've done in Perth that if you've had a PEA arrest to anaphylaxis, the incidence of cognitive dysfunction is sort of 20 or 30%. And we don't know how long that lasts yet. Hopefully it's just transient. But from my point of view, and this is my opinion, there's no greater problem in anaesthesia than anaphylaxis, and that's why I've gone down this route. In Australia, about 3 people die per year, which is 2% of cases of anaphylaxis. And for every person that dies, there's about 2 people that have serious morbidity, so hypoxic brain injury, something like that. So to prove that a test dose given by this system works, I really have to show 4 things. The first one is that anaphylaxis eliciting doses are highly variable, and this is why a single test dose won't work, or at least is less effective, or isn't the ideal solution. Anaphylaxis is a dose-response phenomenon because this is the opportunity to make a difference. The latent period of IV anaphylaxis has to be short, and I think this is why immunologists have never discovered or gone down this route, because they give all of their drugs orally, all their challenges orally, and so the idea of giving a drug in this way hasn't been explored. And the fourth point, which is very important, is that the dose-response range is proportional to sensitivity. And what I mean by that is that if a patient is sensitive, they're sensitive. So if they're very sensitive, the range between the start of the reaction, the dose for a mild reaction and the dose for a severe reaction is very narrow. If you're not very sensitive, the range is very wide. And this means that we can accelerate the infusion along that 30 minutes in a particularly efficient way. So anaphylaxis eliciting doses are variable. They'll follow a bell-shaped curve. This is what I talked about earlier. This is peanut allergy, and this is on the x-axis is the log dose of the allergen, and this goes over 7 orders of magnitude, which is remarkable. It's a huge range in dose. And I mean, log dose is how biological systems work, biological systems don't really work in a linear fashion. So there's a million-fold inter-individual variability in eliciting dose responses for most allergens that you look at. If we look, so that was a cumulative graph. On the y-axis was the cumulative percent of the population that are allergic that would respond at that particular dose. But we can just look at intervals, which I've done on the right-hand side. Now, each one of those intervals is a order of magnitude. So if we were going to give, say, 5% of the dose, that would look like this. So if we give 5% of the dose, we would expect to trigger the vast majority of the population. And that's why I'm so focused on giving very small doses at the start of the infusion. This is the same graph as before, but with the x and the y-axis flipped. So on the x-axis is the cumulative population of responders, and on the y-axis is the cumulative dose, which is in a logarithmic dose. The yellow line is the subjective or the minimal dose. So what they've actually looked at when they've generated these curves is they've looked at subjective and objective, subjective symptoms, objective signs in awake patients being given oral allergens. But if we were to run, say, I used to think that the safest way to give a test dose 10 years ago was to put my antibiotic into a litre of saline or CSL and just run it over half an hour. But if I do that, after 5 minutes, I've exceeded the threshold dose for 70% of the population. And 5 minutes probably isn't long enough to, because of the latent period of the reaction, isn't long enough to recognise the reaction and stop it before you've given too much of a dose. You can create the same curves by using provocation doses from challenge clinics that people have done with oral dosing. And again, you see a millionfold variation in eliciting doses. 20% of the population will react to 0.1 milligrams of penicillin. And this is another curve I've created. I like taking data from papers that wasn't originally intended for a particular use and then reworking it, but I'm already talking over. And you can do this for all kinds of things like quinolones and everything. Chemotherapy agents as well. So what this is a very well-known Mariana Castell's chemotherapy desensitization protocol, and they've looked at which stage of the desensitization protocol people who did have reactions had them, and it's distributed again over many orders of magnitude. And this is why test doses don't work. So, point one, yes, anaphylaxis eliciting doses are highly variable. And there's a great opportunity for researchers to look into anaesthetic drugs, intravenous perioperative drugs, and to see what the actual eliciting doses are, because that would better inform us. Point two, anaphylaxis is a dose-response phenomenon. This is true. We know this is true. The existence of desensitization protocols tells us that it's a dose-response phenomenon. The recommendation that we remove a trigger once a reaction's recognised indicates that it's a dose-response phenomenon, although you might not agree with that. There's a correlation between tryptase levels and severity. There's a correlation between skin test wheal size and concentration of the allergen. In vitro, you can look at basophil activation and different concentrations of the allergen. And over a certain range, it's a dose-response phenomenon. But there'll be arguments about it. So, letter to the editor here, anaphylaxis is not a dose-response phenomenon. And that's true. Both people can be right. It just depends what dose you're giving. So, dose-response curves. The one on the, you can see now I've moved into a non-responsive part of the dose-response curve, the flat part on the right. These responsive parts of the dose-response curves, when you're talking about allergy, are phenomenally, what would you say, they're incredibly potent stimulators of IgE. So it's at very, very, very small doses that you'll stimulate these. You'll notice that the x-axis here is a logarithmic scale. So we're now at a trigger part of the reaction, the minimum eliciting dose. If we can recognise that it's anaphylaxis, we can turn it off before we get to the maximal response. But the question is, in these very tiny doses, are we going to be able to do that? And also, are we going to be able to do it for all 4 of the patients shown here? Now, I said that the x-axis was a logarithmic scale. If you turn it into a linear scale, it does look like a dose-response, looks like an all-or-nothing curve. So the graph on the left is exactly the same as the one on the right, except for it's got a linear x-axis, horizontal axis. And certainly to a constant infusion, that sure as hell looks like an all-or-nothing response. But if we give the dose in a logarithmic way, then it starts to look like a dose-response curve. So let's do that with this. So this is a population of 30 patients, all with different EC50s, which is their dose at which they're sensitive to the drug. And just for simplicity, I've made them all have the same Hill coefficient, which is the slope of the curve, which defines what leeway you have between the minimum and the maximum dose in terms of response. So what we could do is just evenly go through all of those patients one by one. Start from the left and move to the right. Let's see if I can do that again, because I like that graph. It took me forever. So to do that, what you need to do is you need to move across the x-axis in a logarithmic way, because that's what that red line is doing. It's moving at an exponentially greater speed as it goes from left to right. And that's what this graphic on the right shows. So the dotted line is a constant infusion, and it's very small, but what you'll see is on the left of the, I've indicated 0.01% of the cumulative dose, 0.1% and 1%. It all happens in the first 30 seconds of a constant infusion over 30 minutes. And yet I've shown you how many of your patients you will have already triggered, probably maximally in those points. The infusion that I'm proposing is the black line on the right, and it gets to 0.01% at about 4 minutes, it gets to 0.1% at about 10 minutes, it gets to 1% at about 16. You see what I'm doing? I'm separating orders of magnitude by the same distance, and that makes sense for something that's has a proportional response in terms of the reaction. So you can, on the back of an envelope, work out how fast we can give this infusion. We know from penicillin provocation, we know from penicillin desensitization protocols, ultra-rapid protocols over 4 hours, we know that they start at about 0.01% of the total dose. So we can say that anything below that we don't have to worry about. So the threshold for everyone probably starts at about 0.01%, and we go up to 100%. So that's 4 orders of magnitude. The dose-response curve, how wide is it? It's at least 10 times, it might be 100 times. So that tells us how fast we can move through each of these patients. And the other thing we need to know is how fast can we recognise a reaction? What's the latent period for IV anaphylaxis? So the minimum's 0.01%, the dose-response curve, if it's at least 10 times, and if the latent period's at least 5 minutes or less, we can accelerate through these 4 orders of magnitude in half an hour and give everyone about 7 minutes to recognise the reaction and turn the infusion off. Now, there's lots of things that affect the severity of anaphylaxis. And these are listed, all the things you can have, sensitising agents, other factors, exercise, all those sort of things. The only thing we as anaesthetists can change is the dose. I'm going to skip over this because I'm going a bit slow. Last week, I pulled out a whole bunch of files from our clinic, and I just grabbed 20 and 21. 22 and 19 are on my bedroom floor at the moment, haven't been done yet. But there was 46 cases of cefazoline anaphylaxis under anaesthesia. And as you can see, so this is a histogram of the latent period. So the beautiful thing about ANZAC referrals is that they have time the reaction was recognised by the referring clinician, and then they list all the times that each particular drug was given. They don't know which drug caused it. And so over 30%, the cefazoline was given within 2 minutes, over 50% were given within 4 minutes, and over 70% within 6 minutes. So it seems feasible that we can recognise these reactions. And this is a maximum latency. This is when they realised anaphylaxis was happening, when they're doing all their other anaesthetic things. It could be that we can recognise these reactions much sooner, beg your pardon, if we were more vigilant. But this is a worst case scenario. And NAP6 found a similar thing. So if you look at their most common triggers, which were vancomycin, teicoplanin, and coamoxiclav. Again, the majority were discovered within 5 minutes of their trigger. Now, my fourth point is proportional response. Now, I've talked about these cumulative responders data sets, and they show that the difference between say a minimum and a maximum reaction varies, and the variation between those two doses is proportional to what absolute dose you're triggered by. So let's look at cashew nuts. If you were triggered by 1 milligram for your subjective symptoms, your objective sign happened at 100 milligrams. So if you were triggered at 1 milligram, the width of that response curve was 99 milligrams. If you were triggered at 3, it's 300 milligrams. If you're triggered at 10, it's 1,000 milligrams, i.e. it's proportional. And this is very important because it lets us accelerate our infusion. It tells us something. We might not know where you are in terms of your sensitivity, but we do know if you are at that point in sensitivity, we do know how much of a range we have. And we can look at it, I think this is oat something or other, and it's the same sort of thing. And you can do this for lots of different allergens. But again, all the research has been done in food. This is a great opportunity for any researcher to look in the perioperative space. So proportionality of response is important, and it means that a constant infusion is never safer than the proportional infusion. And a single dose is never safer than either of those. So if you want to minimise the risk, we need to use proportional infusions. So we've established the 4 points I set out to establish to give some theoretical basis to using this device. Now, we've already looked at the population inverted, so I'm going to skip over that. Individuals that are allergic, they'll appear like this. And we can bunch them into groups. We can look at people that have different sensitivities in the population. So let's look at this blue line would be, say, the 10th centile in the population, and they would react at a minimum to 0.05%, and they would have a maximum reaction at 0.9% of the cumulative dose. Skip that one. So if you ran a constant infusion, you would exceed their objective or their maximal signs at 27 seconds if you were running a constant infusion over half an hour, which means there's absolutely no time. It's a complete waste of time. You might as well have just given the whole dose as a bolus. If you looked at the 30th centile, the 50th centile, the 70th centile, those widths with the constant infusion get longer, but they're still very narrow. So the 30th centile, you'd have 1.1 minute. The 50th centile, you'd have 2 minutes. It's still not long enough to recognise the reaction and turn off the infusion. When you get to the very few patients that are very non-sensitive, and I might add that they also know that people who aren't very sensitive tend to have less severe reactions. The most severe reactions tend to happen like for like if you're to the patients that are highly sensitive. But once you get to those patients that aren't very sensitive, a constant infusion is starting to look okay, in that you've got 6.5 minutes to recognise the reaction and turn the infusion off. And the way that people have coped with this in the past, this linear infusion, is to stretch their desensitisation protocols over hours. And so Mariana Castells, who's the world leader in this, runs her infusions over 350 minutes, over more than 5 hours, almost 6 hours. And that's impractical. We can't do that, obviously. That's ridiculous. But why do we have to wait? As I've shown you, we can overlap challenges without losing effectiveness. We can compress it into 30 minutes. I'm glad that came up again. So we can do overlapping testers. So with this constant infusion, let's look at the 10th centile. Before we had 27 seconds. Now with this infusion, you've got 8 minutes, 8.2 minutes. So, after 3 or 4 minutes, they might develop high airway pressures or some hives or something. Turn the infusion off, treat that, disaster averted, turn the infusion off, give an alternative antibiotic. And it's you can finish the rest of your list. You don't have to cancel it. The patient doesn't have to have their surgery cancelled, etc. And if you look at all of these centiles, they all have the same safety margin. So we've got 8 minutes for the 10th centile, we've got 7 minutes for the 30th centile, we've got 6 minutes for the 50th, we've got 6 minutes for the 70th. Every possible person with every possible threshold has a safety margin. And I think, yes, this one works. So there you can compare them at the same time. So it's a superior infusion compared to a constant infusion. And, you know, I would have always thought, my God, you give your antibiotics over half an hour as a constant infusion, you must be the most risk-averse anaesthetist ever. Wrong. So that was my idea. It's actually not as easy to put into practice as you might think. So the final infusion rate, in terms of dose, is 25,000 times the initial infusion rate. Sorry, there's a typo here. If you put your antibiotic dose in 50 mils, then in the first minute you've given 1/1000th of a mil. So now you understand why I need a dilution chamber in the syringe. So the actual starting rate's about 27 mils per hour because of the dilution chamber. So that makes this whole thing work. It makes it practical. It makes it actually doable. And also after 30 minutes, you've only given 0.5% of your dose. But that's, as I've shown you, that's what's required for this to work. This is my dad. He's a biochemist. I couldn't have done it without him. So it's quite difficult. These are the engineers we have from Melbourne who started out by using a magnetic stirrer to try and get good mixing. And it was quite hard to get it working. We set up lots of test beds to examine mixing in the dilution chamber. Lots of testing. It's been 3 years of testing, and using dyes and a spectroscope to hone the result. And we've got a US patent. And the big question is this feasible? I don't want to make anaesthesia any harder. I want to make it easier. So the question is, how much of a nuisance is it? I've shown you how easy it is to set up an infusion the second time you've ever done it. Only been introduced to it 5 minutes earlier, which is what Russell was. When do we start it? What about skin incision and dosing? And how much does it cost? Well, in terms of dosing, when we give our antibiotics, we give a massive dose. The reason why we give a massive dose is because we're giving antibiotics as a bolus. So if we give a non-obese person 2 grams of cefazolin, the peak concentration in the blood is 80 mics per mil. The MIC is about 2. So the MIC for E. coli, I mean, for Enterobacter, it's a bit higher, like 8 to 16. So let's just say we want an MIC of 8 to achieve that before skin incision. So we're overdosing by a factor of 20 at least when we give our antibiotics. And the reason for that is because that overdosing pushes the redose interval out to 3 or 4 hours. But that doesn't mean we have to get that whole dose in before skin incision. You can, with this particular algorithm over 30 minutes, you'd only have to run it for 22 minutes to get that initial concentration in. So that's somewhat of a reprieve. And that's with 1 gram of cefazolin. Will it fit into our workflows? Well, there's been some research done on, trying to reduce non-operative time. It's obviously a big focus it's a big focus everywhere to reduce your non-operative time, your induction time, your emergence time, and your turnover time. If you just look at induction and turnover time, that's more than 30 minutes, and this is when they're doing a study to try and reduce turnover time. So I think you should be able to find 22 minutes somewhere, put a drip in, put the patient to sleep, position them, prep and drape the patient. Before skin incision, I think you should be able to find 22 minutes. What's a similar inconvenience in anaesthesia? Pre-oxygenation, awake blocks, awake antibiotics, checking pressure areas. And as I've said before, risk minimisation is a key attribute of the anaesthetist. And in WA, we see about 200 people a year that has had perioperative anaphylaxis. It's quite disruptive for them and their families. And perhaps the role for this thing isn't in theatre at all. Maybe it's for the wards, or maybe it's just for people who are allergic to your first-line agent. So, for instance, if you're allergic to, as I've said, if you're allergic to penicillin and you want to give them a beta-lactam, and you know that their rate of anaphylaxis. So if you have a self-reported penicillin allergy, their risk of reacting to cefazoline is probably about 1%, 1 in 100, so it's probably worthwhile in that 10% of patients using something like this so that you can recognise the reaction. And you might say, well, why would I take that risk? Well, if you don't give them cefazoline, you're going to double their surgical site infection risk. And actually giving them an alternative gives them about the same or even higher risk of anaphylaxis anyway. So just give them cefazoline, but use a test dosing device like this. The cost of a dose of cefazoline, it's cheap, it's cents. But the cost of anaphylaxis to cefazoline is a lot. When they did COVID modelling, they modelled giving the COVID vaccine on the basis that it was worthwhile if you spent $10 million to save one life. So if the cost of if the cost of a death from anaphylaxis is $10 million, then that's $22 per dose of cefazoline. If the cost of the invention is $20 per unit, then that's an overall cost saving to the health system. And I imagine we should be able to produce these at $1 or $2 per syringe because that's the only disposable bit. So it's more about the absolute focus has been trying to design something that isn't a pain in the arse to use. So there's lots of potential for research. I've applied for 16 grants so far and I've been successful in 0 of them. The evidence for everything I've said exists, but it's all incidental evidence. The research has been done for other reasons, and I've just sort of photocopied their papers and reanalysed their data. But there's lots of opportunities for us to do research to define the drug provocation threshold for our drugs, to use these kind of devices in challenge clinics, because the beauty of it is if you run this syringe and after 4 minutes the person has a reaction and you turn it off, you know exactly what dose you've given at that point. You know, you've given 0.02 milligrams of cefazoline. If you run it for 6 minutes, you've given 0.7 milligrams of cefazoline. So we'll be able to start to create a database of dose provocation thresholds. And maybe that might tell us in the future that we can accelerate this part of the infusion or that part of the infusion and make it over 20 minutes or 10 minutes. Or maybe it needs to be longer, 40 minutes or 50 minutes. There's other places we could use it. Iron infusions is an ideal place. If you could use, I think it's iron, I can't remember which is the cheap one now. Yeah, we could use polymaltose instead of carboxymaltose. That would save, be $50 versus $300 for each dose. So there's a commercial argument from the first dose. So the other thing you'll notice when you go, I actually went and chatted to one of the nurses on the ward because I wanted to see if they'd ever used this. I said, what do you do when you're giving an antibiotic on the ward? And they said, oh, well, look, we just sit with the patient for the first 5 minutes. And you can understand completely why they only bother sitting with them for the first 5 minutes because that's when the reaction happens. And that's it. Thanks very much for your time.
Lovely. Thank you, Paul. I would like to introduce the other members of our expert panel here today. So Dr. Helen Crilly is a specialist anaesthetist at the Tweed Hospital on the border of Queensland and New South Wales in mixed public and private practice. She's been involved in the management and investigation of patients with perioperative allergy since 2002. Helen is a founding member of the Australian and New Zealand Anaesthetic Allergy Group, ANZAG, and she has been involved with many ANZAG roles since 2010, including the ANZAG website, management and testing guidelines. She considers herself privileged to have been part of a group that have always been focused on peer support and patient safety. Above all, she's grateful for the friends and valued colleagues that have been found through ANZAG over the years. Dr. Sarah Green is an anaesthetist currently practising in Sydney in both public and private settings. She's been closely involved with the perioperative allergy service at Royal North Shore Hospital since 2011 and is the current director, leading the team in their objective to provide excellent patient care. Sarah is the Vice Chair of the ANZCA CPD Committee and affiliated to the University of Sydney as a clinical lecturer. Enthusiastic about patient care and interaction, Dr. Green finds the diversity of patients she encounters each day to be a constant source of interest and motivation. And then lastly, Dr. Jamalee is a clinical immunologist, allergist, and immunopathologist from Sydney. She is actively involved in the multidisciplinary anaesthetic allergy service at Royal North Shore Hospital. She is the current immunology representative for ANZCA and a member of the Drug Allergy Stream Working Group for the National Allergy Centre of Excellence. Her MPhil was based on in vitro testing for neuromuscular blocking agent anaphylaxis. She continues to be an active clinician researcher with a focus on antibiotic and radiocontrast allergy, with ongoing publications in these areas in peer-reviewed journals. The panel today invite you to ask your challenging questions on perioperative allergy. Okay, so if anyone would like, I'll start with a couple of questions that we have here already, but if anyone would like to ask a question on the floor, if you just come up to the microphone, then we'll come to you. So just a couple of questions for Paul first, following that excellent, it was really interesting talk. It was very interesting. But a lot of people are asking that we don't always have 30 minutes, and I know you pared that down to 20 or 22 minutes or something. We don't always have 30 minutes of monitored care time prior to theatre. How do you get around the potential theatre delays?
Yeah, well, I mean, it's a system. It'll be a system thing. So if we decide that this is beneficial, then the systems will have to change. The thing about the best monitor for anaphylaxis is an awake patient. So if you were to start these infusions in an awake patient, I think you wouldn't necessarily, you'd have to have a patient in an area where there's a clinician, but I don't think they'd have to be monitored because when we do drug challenges, almost always, the patient will tell us that they're feeling something a long time before there's any objective signs. So I think the best monitor is an awake patient. So that would be one possibility, in that you would, in the doser areas, start your antibiotics there. The recommendation is, if you look at any perioperative antibiotic dosing protocol, is for the antibiotics to be started half an hour before surgical incision. There's nothing really magical about, I mean, if you give an antibiotic 30 seconds before skin incision, I don't think it magically gets to the point that it needs to be. But yeah, look, the answer is the same as, I mean, in my institutions, our turnovers are never under 30 minutes, and that's a system problem. So we need to fix the system just to be more efficient anyway.
Okay. This question's been asked in a couple of ways, so I'll try and put it together for everybody who's asked it. If you're test dosing sort of every patient that's reporting a potential allergy, how many cases will you need to treat? How many will you need to do to prevent one case of anaphylaxis, do you think? And if that would be if we're just looking at cefazolin, for example, and could you use this for other drugs like muscle relaxants?
I mean, you actually, you could use it for muscle relaxants in some sense. I mean, obviously not for rapid signal. Well, I mean, you can give awake. So when we do muscle relaxant, well, no, what I mean is we give muscle relaxant. We do give muscle relaxants to awake people in the challenge clinic. So we usually give about up to 5% of the dose. Or we always give 5% of the dose. We usually give it over 10 minutes as an infusion. Well, there's various different protocols, but that's how we do it. And before we do it, we tell the patient, look, you'll get some blurred vision. And they do, usually they get some blurred vision, but otherwise they feel fine unless they have anaphylaxis. So if you go, so you could potentially use this to give the sort of 3.5 orders of magnitude up to 5%, and then stop it. And you would exclude, or you would identify, more than half of the patients with muscle relaxant anaphylaxis. But, that sounds like a bit of a fiddle, and I would never push. I mean, I just think this would be ideal to start with using it for antibiotics. And I think initially I would start to use it for just patients that have a self-reported penicillin allergy, because there's a very good argument for giving those patients a cephalosporin, and they're at high risk of anaphylaxis. So, yeah, I wouldn't say give this to everyone. I would just say give it to the 10% of your patients that are allergic to penicillin or report that they are. That would be a good place to start. And if I was on the ward, anywhere on the ward that you're giving constant infusions, they should just use this because you lose nothing and it's safer.
Just to follow up on Paul's comment, I think as an immunologist, we stratify the allergy label to start with. So, if the first thing we do is a label no risk, that's, for example, a patient with a penicillin allergy who says they've got a penicillin allergy because their mother has penicillin allergy. So there's even an argument across many sort of professionals that you don't even need to challenge, but often the patient will benefit from one because then they know that they can actually take it. Then, then you've got the low risk labels, which could be, from in an anaesthetic setting, someone who's got a skin test negative reaction to a drug, and you might be able to classify as low risk, and therefore I wouldn't really go down the test dose or graded challenge path, and you probably just give a full therapeutic dose. But then the most difficult bracket is the everything else, the non-low risk category. I guess it's hard for anaesthetists to work out sometimes which category it is. So for us, non-low risk categories I think is where the challenge is. Some labels that you might think are non-low risk are actually probably not. For instance, someone with a label of an antibiotic allergy that is anaphylaxis, like real true anaphylaxis, but it was 15, 20 years ago. Even though the label is non-low risk, because of the elapsed time, it actually becomes a lower risk label. So it'd be really nice. It would actually make the process a lot more efficient for you if you had the time, to really delve into the label and work out sort of what the risk stratification is before proceeding, because it would save a lot of time.
But it'd probably be faster just to use the infusion.
We don't have the luxury of having a perfect setup for IV challenges. We have to operate out of the outpatient service, so there's a lot that we're not allowed to do there.
We have a question from the floor.
Hi, Karen Pedersen from Auckland, and I work in the anaesthetic allergy clinic there. I think it's really important to differentiate between giving penicillin to a patient with a penicillin allergy history, in which case those risk stratifications and detailed history are really important, as opposed to giving cefazolin to a patient with a penicillin allergy history. And I think we're all aware that there's growing evidence that actually our historical 10% risk of cross-sensitivity between penicillins and cephalosporins doesn't apply to cefazolin because of its unique side chains. So in fact, when we went back and audited our cefazolin anaphylaxis cases, the vast majority of people who have anaphylaxis to cefazolin have got no history of allergy to penicillin. I don't think that a history of allergy to penicillin necessarily identifies a high-risk group of patients for a history of allergy to cefazolin. So, I think we need to be very clear that those are not the same things.
Yeah.
Giving penicillin to a penicillin allergic patient is very different to giving cefazolin to a penicillin allergic patient.
Yeah, I think that's a great comment. In fact, I don't know if Alison, you can skip forward a few slides to.
Yes.
Prepared one slide on this, I think.
Yes, here we go. Do you want the whole thing up?
Yeah, I think you just pull the whole thing up. Yeah, I think that's the first point. I mean, just to really summarise, I really think that the risk of having a cefazolin allergy in a patient with any level of penicillin allergy is way less than 1%. And even in patients with true penicillin allergy, it's probably less than 1%. So I think it's a very no to low risk scenario for us. So I guess the first point we've already talked about, first thing, the penicillin allergy label, chances are it's not a true penicillin allergy anyway. So that just reduces your risk down by a whole lot. The second point there I've written, so for the body to recognize a penicillin or a cephalosporin and produce, anaphylaxis or a milder drug reaction, it requires the drug to be haptonated, which is, a process where it just becomes becomes a different molecule and conjugates to certain residues. Penicillin allergy, we understand how that works pretty well. The process of the haptonation, which parts of the penicillin itself, attached to which residues. Cephalosporins, we don't really understand as well. And the process for haptonation of cephalosporins is actually not very well understood. So I guess where that gets to is that the mechanism of penicillin allergy is actually quite different to the mechanism of cephalosporin allergy, which then also, adds to that. They're two sort of different separate processes, and that the cross-reactivity is way, way, way lower than 10%. I think historically they probably thought it was more similar process, and therefore got to that value, but I think it's pretty clear now that's not the case. Maybe if you can go to the next slide. So there are some select instances of penicillins and cephalosporins that we do worry about, and the ones I've circled in red are the ones that are, available in Australia. So for penicillins to cross-react with cephalosporins, like be more than a low-risk scenario, you do have to have that sort of similarity in the side chain. And that comes mainly between ampicillin and cefaclor, and amoxicillin and ampicillin and cefalexin. So, and that's not all the time either. I think the quoted figure in some publications back in the day was about 25%. In our hospital, we don't really, or even in this state on an outpatient basis, we don't really use ampicillin all that much. We use amoxicillin. We also prescribe a lot of cefalexin and cefaclor. My personal feeling is that the chances of someone with true amoxicillin allergy having an issue with cefaclor and cefalexin is 10%. That's just from our sort of data collected at our centre. So if you see cefazolin, which I've put in red way down in, I think, group 6 or 7, it doesn't even share those similarities in the side chain. So it's, it's definitely less than 1%.
Just while you're there, Gemma, is there such a thing as beta-lactam allergy?
It's not a term that we like to use anymore because it's just so heterogeneous as to what we're talking about. So, in our drug allergy letters, we should definitely refer to the drug itself.
And just before we move on from Paul's topics, so that other people get a chance to talk, there's several people asking about when you might not use your system, sort of who not to use it for, but particularly what about caesarean sections, for example? Would those patients be appropriate to use it for?
I think that would be the ideal patient to use it for.
What about and emergencies in particular?
Well, look, I think if you don't have time to use it, you don't have time to use it. If I would, for instance, if I had set it up and started the infusion and the surgeon was, looked like he was going to make his cut in 2 minutes, I'd just take the syringe out of the driver and just give it as a bolus like you normally do. I mean, it's not about risk zeroing, it's just about risk minimising. If you've got the time to give it, if you've got the time to use it, just use it. You haven't lost anything. If you find you don't have time, you just give the antibiotics because most likely the most important thing for that particular patient is to get the antibiotic. The other point is that if you've got a self-reported penicillin allergy, your mortality, your all-cause, they followed up people for 6 years with self-reported penicillin allergies. Your all-cause mortality was 14% higher. They've done studies where they look at your perioperative infection rate, and it was double, and that was entirely due to your choice of antibiotics. It wasn't the fact that you're identifying people with a higher risk of infection because they've got allergies. It wasn't like they had weeping eczema and that's why they got infected. It was 100%